So that will take us into the neuron. Now we've already discussed the structure of the neuron. We have the cell body and we have the projections that come out of our cell body. We have the axons and the dendrites. Now the axon is the major structure, the very long projection that sends signals out to other cells. And the dendrites are all of the littler projections that receive signals and consolidate different messages from different other cells coming into our neuron. But we have not discussed the characteristics of neurons. The characteristics of neurons are very, very unique to this type of cell. So one of the characteristics that we're already aware of is that our neurons are what are known as electrically excitable. If you guys can remember, our skeletal muscle cells were the same way. Those were electrically excitable, which just means that these cells can send action potentials, which are just signals sent from one cell to another. But some of these other characteristics here are ones that we have not heard so much about. So I'm going to take some time to go over each of these. So first and foremost, our neurons are amitotic, which means that they do not undergo mitosis and cannot undergo cell division. Now this is the current scientific standard about neurons at the current time. However, there's a lot of research going into neurons, actually looking into the fact whether or not these are able to divide. And there are certain researchers looking into the possibility of stimulating neurons to divide. The medical implications of stimulating neurons to divide is repairing damaged nervous tissue, which is not something that the body can do very well regularly. So this is currently a big area of research, is trying to find ways that we can stimulate our neurons to have mitosis and make more neurons. We can make more neurons, we can replace neurons that have been damaged. So, at the given time, this is being debated and we are researching into topics like this. However, if this does show up on the test, our neurons are amitotic, or in my class, they will not be able to divide. But I just want you to be aware that there is a lot of research going into this right now. Now, our neurons are amitotic, so they're not able to make new neurons. However, what our neurons are very, very good at is being plastic. Our neurons are very malleable, so we call that neuroplasticity. So while our neurons can't necessarily divide to make new neurons, what they can do is sprout new projections and make new processes out of their cell body, which can attach to other cells, and that can make new connections. And this is how we learn. We make new connections. As you guys watch this video, your brains are hopefully making new connections to associate certain facts with others. So this is how we learn. Now this is also the way that we unlearn things. We can disconnect certain pathways that haven't been used for a while, and that's going to decrease the likelihood that we're going to send a signal down a certain pathway. So neurons are plastic, which means they can't necessarily make new neurons, but they can sprout new projections, make new connections, and that will in turn help us to learn new things and increase the efficiency of how we learn. So this is a really big characteristic of neurons. So this is a really, really big concept about neurons. Neuroplasticity is a very important characteristic. This gives our neurons the ability to learn new things. Now the two other major characteristics we see here, longevity and high metabolic rate. A high metabolic rate just means that our neurons need a lot of oxygen at all times. Our neurons cannot go without oxygen for very long, or it will cause damage to our neurons. And this is why when blood flow stops to the brain, that can cause a lot of damage to the nervous tissue that's found within our nervous system. So these cells require lots and lots of oxygen. Other cells in our bodies can go without oxygen for a little bit longer, but these cells are very sensitive and they'll need a lot of oxygen all the time. Now, longevity just simply means that our neurons are very, very long. Neurons can be very, very short, and they can have a very short axon that reaches from one location to another. But certain neurons that can be found in the large toe actually reach their axons all the way up to the brain. So neurons can be up to 6 feet. And that's what we mean when we say longevity. Neurons just simply are very, very long cells because their axons need to be long in order to reach faraway places. So these are the major characteristics and the structures of the neurons. Again, we've discussed the structure of the neuron quite a bit, and we will continue to discuss the neuronal structure in this presentation. But these characteristics are very, very special to our neuronal cells, and some of these characteristics are what help our neurons have its communicative function, which is the overall function of our neurons in general. So up until this point, we know that neurons have dendrites, and we know that neurons have axons. Axons are the large projection, dendrites are the multiple smaller projections. And we know that the axons send signals out from the neuronal cell body, and we know that the dendrites will receive signals. But in the central nervous system versus the peripheral nervous system, we will actually call these different structures different things. So oftentimes our neurons exist within bunches. We'll have a bunch of cell bodies that are all grouped together in one area, And then we'll have all of the axons that are running together in some type of pathway. So in the central nervous system, we call collections of cell bodies nuclei, and in the peripheral nervous system, we call collections of cell body ganglia. So we'll see certain structures that are named nuclei in chapter 14, and we'll talk quite a bit about ganglia when we get to chapter 15. But all these referred to are collections of neuronal cell bodies, and they're just named based on where they are found, in the peripheral nervous system or the central nervous system. Now, we also can name the axons. A lot of the axons will all run together in certain pathways, and in the central nervous system, we'll call those tracts, and in the peripheral nervous system, we call those nerves. So all of those peripheral nerves that we see making up the peripheral nervous system are just bunches of axons that are coursing throughout our bodies. That's all our peripheral nerves are. It's just a bunch of axons. Now, every neuron is going to have a nucleus and some other organelles that are relatively common throughout our cells. However, neurons are specialized because of their axons and their dendrites. So you can see the dendrites projecting out from the cell body of this neuron. And they go in all different directions, and they help a neuron receive signals from all different directions as well. But at this part of our neuronal cell body, we have an enlargement, and then that's going to slim up to become this axon. This region at the beginning of the axon is referred to as the axon hillock. And this is actually going to be the region where action potentials are generated. Once an action potential has been generated, it's going to get sent down the neuronal axon, and that's going to eventually go all the way to the axon terminals, where our neuron can now attach to other cells. So you can see the axon terminals and the end bulbs at each of these. You can see the axon branches into lots of different axon terminals. And so if this was a somatic motor neuron, for example, we would have an individual muscle fiber attached to each one of these axon terminals, and that would be a motor unit. We can remember that from chapter 10. Now, along the axon, we can see all of these different Schwann cells that are wrapping around our axon, and they form the insulated myelin sheath, and we discussed that in the last video. Our Schwann cells are found in the peripheral nervous system, wrapping around the neuronal axons, and that insulates the signal that goes down the neuronal axon. So this neuron specifically would belong to the peripheral nervous system because they will be Schwann cells that are wrapping around here. If this neuron belonged to the central nervous system, we would not see Schwann cells, we would see oligodendrocytes instead. And you can see that along the axon we have the axolemma, which is just the plasma membrane of the axon, and along the Schwann cells we have the neurolemma. Now you can see that as we slice through this Schwann cell, we can just see this one cell that has sort of made its way around the axon. It's wrapped around in many different layers, and so we call this a fatty insulator because this is just a bunch of layers of phospholipids that are wrapping around our axon in this area. Now, something that you also might have realized about this picture is that between the myelin sheaths, or between the Schwann cells that make up the myelin sheaths, we have these little bare areas right in between, and we call these nodes of Ranvier. Those are the nodes of Ranvier. And so the way that this signal is getting sent down the axon, these Schwann cells are insulating the axon in these areas, and it almost appears as if the signal jumps from node to node. So the myelin sheath is truly helping our axons send signals much, much faster. That's what myelination does. When we myelinate our axons, we can send signals much, much faster. So you can see a little neuron here. Here are the dendrites, and here is the axon. We can see the dendrites branching in all different directions, so this neuron can receive signals from all different neurons that are in all different directions. And then once that neuron decides what signal it wants to send, it's going to send that right down the axon. So the dendrites are a lot shorter than the axon, and they're going to be a little bit more diffuse. They're going to be found all over our neuronal cell body, and we'll just have this one long axon. And so this is how you can tell the difference between the two. The axon is going to be a lot longer than our dendrites will. So every dendrite is going to have little spines known as dendritic spines, and those will be what receive the signals. And they're going to pass on those signals to the cell bodies in the form of what is known as graded potentials. Graded potentials are smaller signals that will travel from our dendrites to our cell body, and then those will all summate to form an action potential fired at the axon hillock of our axon. So this picture is a nice way for us to look how two neurons might communicate with each other. You can see that this one neuron is going to project its axon out, and there's going to be a lot of axon terminals that are found all over the dendrites and the cell body of this second neuron. So every neuron is either a presynaptic neuron or a postsynaptic neuron. Remember, in Chapter 10 we discussed the synapse. Well, here in Chapter 12, a synapse is anywhere where two cells come together. That's all a synapse is when one neuron binds to another cell. We're going to have that synapse. And with the muscular system, it was just called the neuromuscular junction. That's a special synapse that we see. However, more commonly in this chapter, we're just going to refer to those locations where two cells come together as synapses. So you can see that the presynaptic neuron is the one that sends the signals out. This neuron will synapse on the postsynaptic neuron, and then our postsynaptic neuron will send signals out to other places. And then if there was another neuron over here, for this signal, this would then become the presynaptic neuron, and whatever neuron's found over here is going to be the postsynaptic neuron for that signal. So neurons exist within circuits, and we just use these two terms to help us understand which cell is sending signals and which one is receiving signals. The presynaptic neuron is sending signals to the postsynaptic neuron, and the postsynaptic neuron is receiving signals from the presynaptic neuron. So again, you can see the short diffuse branching of the dendrites. You can see our cell body, our axon hillock, where our axon is going to begin. You can see the axon shown here. The myelin sheaths are not shown in this example, but they will be wrapped around this axon. And then where our synaptic end bulbs will attach with another cell, we will see our synapse. And this looks a lot similar to the one that we saw in Chapter 10, because all synapses will pretty much look very similar. So the axon will eventually branch, and we call those terminal branches. Now, the amount of branches is going to differ with different types of neurons, but you can see here that 10,000 terminal branches is not an uncommon number of axon terminals for us to see coming out from one axon. So that's quite a bit of terminal branches. Imagine if we were looking at the neuromuscular junction. We'd have one neuron controlling 10,000 muscle fibers. That is going to be a pretty large motor unit. And then the axon terminals themselves are these little bulb-like structures that will eventually release their neurotransmitters into the synapse. So our action potential is going to get fired down the axon hillock, it's going to travel down the axon, and it's going to separate into all of the axon terminals and produce some type of effect on the postsynaptic cell. And just as we saw with muscles, we have very long cylindrically shaped muscle cells, and we call those muscle fibers. Nerve fibers are just very, very long axons. Those are just very, very long cells, and that's why we call them nerve fibers, because they resemble a fiber structure, because they're very, very long. So by now I've said many times that with axons, we generate that action potential in the axon hillock, and that's going to get sent out from our cell body down the axon. And when that signal reaches the axon terminals, we're going to have the release of neurotransmitters just like we saw in Chapter 10 with excitation-contraction coupling. Now I just want you to be aware, in Chapter 10 we just learned a very specific synapse between a muscle cell and a nerve cell. Now in Chapter 12 we're just going to apply our understanding to two different neurons that have been attached to each other in some way. So a lot of the understanding that you guys gained from Chapter 10 is going to be translatable to Chapter 12 as well. Now this slide is pretty much just to reiterate what I've said about the myelin sheaths so far. In the central nervous system, they are formed by oligodendrocytes, and in the peripheral nervous system, they're formed by Schwann cells. And you can nicely see in this picture, this Schwann cell, so this is going to be in the peripheral nervous system, you can see the Schwann cell taking its cell membrane and wrapping around the axon of our neuron, and it wraps and wraps and wraps until it has this very, very extensive myelin sheath that's formed around the axon. And so we call this a fatty insulator because this is wrapped in the cell membrane of our Schwann cell. And the cell membrane is just a bunch of phospholipids. That's why we call this a fatty insulator. Now the myelin sheath, as I also mentioned before, increases our transmission speed of our action potential that was fired down the axon. That's the purpose of the myelin sheath. That's its main function, is to increase the transmission speed of an action potential down a neuron's axon. Now we can have axons that are unmyelinated. However, we're not really going to discuss them that much in this class. I just got this picture up here that can show you that one Schwann cell will wrap around many different axons in this example, but we're really not going to talk too much about unmyelinated axons. Most of the axons we'll discuss are going to be the myelinated type that we see right here. And just make sure you don't forget about the nodes of Ranvier, which can be found between each of the myelin sheaths formed by each Schwann cell. Now things are just a little bit different in the central nervous system. Our major cell are going to be the oligodendrocytes. Now these are projecting along many different cell axons, And we'll see these form myelin sheaths around many different axons. Sometimes as much as 60 different axons will be coming from an oligodendrocyte. Now this is not really a major distinction, but the nodes of Ranvier tend to be a little bit more widely spaced in our central nervous system. However, the thing that I want you guys to understand the most about this slide is the difference between white matter and gray matter. Now white matter and gray matter are named simply because of how they look macroscopically. White matter appears more white, gray matter appears more gray. So what is the difference between both of these two structures? Well, white matter refers to areas that have a lot more myelination. White matter refers to myelinated axons. So these tend to be different tracks that we will find in our central nervous system. Gray matter, on the other hand, refers to our neuronal cell bodies. So areas of this gray matter are areas where there's a lot of neuronal cell body. And then areas that become more whitish in color are going to have mostly myelinated axons. So I'll show you some pictures of white matter and gray matter as we go through the nervous system. But I just want you to start understanding the difference between white matter and gray matter. White matter is myelinated axons. Gray matter is neuronal cell bodies. And there's not going to be much myelination found in these regions. So we can see on this slide that gray matter refers to our neuronal cell bodies and white matter refers to myelinated axons. On the left we have a more animated version, and on the right you can see we actually have a real specimen. So why don't we go ahead and look at the real specimen. One thing that we can clearly see is that towards the inside of the brain here, we have this very, very whitish looking tissue. And on the outside we have a little bit more, and on the outside we have a darker looking tissue that can be found out here. So these are exactly how they sound. This inside is going to be white matter, and this wrapping around the outside of our brain is going to be gray matter. We can also see certain regions on the inside of the brain that do have gray matter as well, and we can see white matter coursing through certain structures. Now, if we look at the spinal cord, this is a section of the spinal cord, we can see gray matter found on the inside in here, and we can see white matter found on the outside. So this is white matter out here, and gray matter is going to be found out here. So this is actually a distinction that I want you guys to be able to make, is that in the brain, the white matter is actually found on the inside, and the gray matter is found on the outside. And that's different with the spinal cord. With the spinal cord, the gray matter is found on the inside, and the white matter is found on the outside. So let's go over here to our animated picture and see what this looks like. You can see this gray matter on the outside, this line of gray matter coursing along the outside of the brain. That's going to be on the outside, and there's some found on the inside, but we mostly will find gray matter on the outside of the brain and white matter on the inside. That's telling us that wrapping around the outside of the brain, this outermost layer, is where we will find a bunch of neuronal cell bodies. And when we look into this white matter area, this is going to show us myelinated axons. That's going to be the exact opposite when we get into the spinal cord, where we see this layer of white matter on the outside and this gray matter on the inside. So I'll mention this a few more times as we go through the nervous system. But I want you to start making this association and understanding that our neuronal cell bodies are found on the outside of the brain and on the inside of our spinal cord. And our axons are going to be found on the inside of the brain and on the outside of our spinal cord. So next we're going to discuss the structural and functional classifications that we can give to our neurons, and then we're going to enter the concepts of the action potential. So I have alluded to the action potential in lots of my previous lecture videos, but in this lecture video we're actually going to discuss what the action potential is and how it works. But first we need to discuss our neurons. So we'll start with the structural classifications on our neurons. Now our structural classifications and our functional classifications are quite different. Structural classifications refer to what do the neurons look like? What can we see with the neurons? Just as we structurally classify joints. So our structural classification of neurons is what do the neurons look like? Just as we saw with joints, as what is present at the joint, we ask what is present at the neuron. And our functional classification is how does that neuron work? What kind of information is that neuron carrying? So the structural classifications of our neurons is based on what we see at the neuron. What does the neuron look like? This is the same concept as we did with joints. What was present at the joint? What types of tissues were present at the joint? With our neurons, it's what does the neuron look like? So specifically with our structural classification of neurons, we use the number of processes that extend from the cell body to define our different neurons. So we have three types. We have the multipolar neurons, we have the bipolar neurons, and we have the unipolar neurons. So multipolar neurons are named because they have many different projections that come out of their cell body. There's lots of different processes, lots of dendrites and one long axon. With a bipolar neuron, we're going to have two major projections out of our cell body. We can see that all of the dendrites will converge on this one spot, and they will eventually bring all of their signals into the cell body through this one point here. And then our neuron is going to send its signal out of the axon. So this is bipolar because there's two major processes that project from our neuronal cell body. And lastly, our unipolar neurons are going to have a single projection that are coming out from the cell body. So you can see that the dendrites will all converge just like they did with the bipolar neurons, but these are going to eventually come into the cell body through one process that extends from the cell body. And then when our cell's ready to send signals out, it's going to use that same little point to send its signals out through our axon. So in terms of structurally classifying neurons, we have lots of projections, two projections, and one projection for multipolar neurons, bipolar neurons, and unipolar neurons, respectively. Most of the neurons that we've seen up until this point are the multipolar neurons. We'll discuss these the most. And we'll also see some unipolar neurons carrying sensory information very often. Our unipolar neurons most often carry sensory information. Bipolar neurons we see with our special senses, which we'll cover in Chapter 17. So I won't be talking about much more of these in this presentation, but our multipolar neurons and our unipolar neurons are going to be the types that we're really going to discuss throughout the rest of this video. So we've actually already discussed the functional classification of our neurons when we discussed the function of the nervous system in general. Remember, I said that afferent neurons take signals up to the central nervous system, and efferent neurons take signals out from the central nervous system. But we also have this other group, which are known as the interneurons. These are our association neurons that will link sensory and motor neurons together. So sensory neurons come in and synapse on interneurons, and interneurons will receive that sensory signal. And the interneurons are the cells that understand the sensory information. So it receives that sensory information, it understands that sensory information, and then it's going to further synapse on a motor neuron, and it will then tell that motor neuron what signals to send out. So the interneurons are found located between sensory and motor neurons, and it helps us to link sensory signals coming in with motor signals going out. So you can see that most of the neurons that we find in our bodies are actually multipolar. Most of our motor neurons are multipolar, and most of our interneurons are multipolar. However, with our sensory neurons, we will often see these classified as unipolar. These are unipolar because their cell bodies will often sit in ganglia that we will find in the peripheral nervous system. And its dendrites will end up bringing in information and shuttle that information into the cell body. And then the cell body, once it consolidates all of those sensory signals, can send another action potential out of its axon through that same projection. So we'll often see our unipolar neurons representing sensory neurons, and we'll see multipolar neurons most everywhere else. Again, we'll often see some bipolar neurons found in the eyes and in certain structures that help with our olfactory sense, but we will further discuss those in Chapter 17. So this picture shows not only the functional classifications of our neurons, but the structural classifications of our neurons and which nervous system each of these neurons will belong to. So we'll start here at our sensory receptors of our sensory neuron. So our sensory or afferent neurons are going to have some type of receptor that allows them to bring signals up to the cell body. Now these sensory neurons are mostly going to be unipolar, which means that they have one projection coming out from their cell body. So these signals are going to come up into this cell body, and once the cell body decides what signal it wants to send out, it's going to send that out of the same projection up into the central nervous system. So this sensory neuron is going to synapse with certain cells up in the central nervous system, and that's where we will find our interneurons. These typically tend to be multipolar, and we can see lots of different projections coming out of our cell body, so that's going to indicate that this neuron is multipolar. And this is also an interneuron. So this is how our interneurons are going to work. We can see that the sensory neuron comes in and synapses on our interneuron, and then the interneuron is going to link the sensory and the motor neurons together. So when that sensory information comes in, this neuron is simply relaying the signals that it got from its sensory receptors up to the interneuron. And the interneuron is the association neuron that can help make decisions based on that sensory information that came in from our sensory neuron. So the interneuron is then going to send further signals out to the motor neuron once the interneuron has decided what signal it wants to send out. So the interneuron is going to tell the motor neuron what signals it needs to send out and how it should use its different effectors. So a good example of how we will see this in play in the body is perhaps if we were to step on something sharp. I believe I used this example in our last video. If we stepped on something very, very sharp, like a nail or perhaps a Lego, those of you that have Legos sitting around your house know that there is nothing worse than stepping on a Lego. So let's say we step on a Lego, and that's going to be sensed by these sensory receptors. That's going to send that signal up to our cell body of our sensory neuron, and then this neuron is going to relay that signal up to the interneuron. And the interneuron is the first cell that receives that signal and realizes, ouch, that hurts, that's painful, and we need to make sure that we get our foot off of that Lego. So the interneuron is going to tell the motor neuron to contract certain muscles in the leg to pull our leg away from that sharp object. And then the motor neuron itself is going to send those signals out to the muscles of the legs, and that's going to pull our leg off of that Lego. So I like this slide a lot because it combines our structural classifications of neurons with our functional classifications of our neurons. And we can see this arc by which our sensory information is going to come in and eventually be used to produce some type of motor signal that gets sent out. So that will take us into the action potential. Action potentials are just signals that can be sent from cell to cell. And there's actually two versions of signals that our cells can use. We have action potentials or graded potentials. Action potentials can be sent over a short distance or a long distance, while graded potentials can only be sent over a short distance. We'll see action potentials arising in the axon hillock and firing down an axon, and when those signals hit the postsynaptic cell, those are going to form graded potentials, and those will travel over a short distance. So a good rule of thumb is that the graded potentials will travel from the dendrites all the way to the cell body, and then from our axon hillock, we're going to fire our action potential, and however long our neuron's axon is how long that action potential is going to travel. So on this picture, we can see this arc of sensory neurons sending signals up to interneurons, and then those interneurons sending signals back down through motor neurons. So we'll see that graded potentials can be shown with a little blue arrow, and nerve action potentials can be shown with a black arrow. So you can see graded potentials fired in the sensory receptors, and those are going to end up getting sent up through an action potential up to the cell body of our sensory neuron, and then we're going to continue that signal up all the way to our interneuron. You can see that the sensory neuron is going to trigger graded potentials in the interneuron, and then those signals are going to end up in the axon hillock and fire an action potential from the interneuron up to other interneurons. But every time we see a cell body and some dendrites that are synapsing on it, we'll find this graded potential, and then down every axon, we'll see the action potential. So eventually, these interneurons are going to synapse with more interneurons. So there can be more than one interneuron. But eventually what's going to happen is that those interneurons are going to synapse on the upper motor neurons. We'll have graded potentials coming through their cell bodies, and then more action potentials are going to come out. This will synapse on a lower motor neuron, which will trigger graded potentials in the cell body of our lower motor neuron. And then the lower motor neuron is going to send a final action potential out through its axon and take that signal to the effectors, where we can have some type of response to our sensory stimuli. So this is a nice picture to show us the full arc of sensory neurons synapsing with interneurons, and those interneurons synapsing with other interneurons and motor neurons. And we can also see the difference between graded potentials and action potentials on this picture as well. Action potentials can be fired over a long distance, and those are down the axon of our neurons, and graded potentials will often travel from the dendrites all the way to the cell body. So graded potentials and action potentials are very similar. Action potentials are just able to be fired over a much longer distance. So both of these signals are going to arise very similarly, and we're going to have the same criteria needed in order to fire each of them. Now, the two major criteria required in order to fire an action potential is a resting membrane potential and certain ion channels. Now, we've talked quite a bit about ion channels up until this point, but we have not really mentioned membrane potential. So let me touch on each of these channels real quick, and then we'll make our way into the electrochemical gradient, and then I'll explain what membrane potential is. So there are lots of different types of membrane channels. We learned a lot about different channels versus different carrier proteins for cellular transport through the membrane. However, there are different types of channels, and those channels can work in different ways. We have passive channels and we have active channels. Passive channels can be called leak channels because these channels are always going to be open. They're always allowing their substances to flow through the channel. The active channels are going to be referred to as the gated channel. Now, I briefly mentioned that channels can be gated in one of my earlier videos, but there are different types of stimuli that can open or close these different gates. So we can have chemically gated, which are ligand-gated channels. These are some type of channel that opens in response to a specific chemical or neurotransmitter being present at the gate. There will be a receptor on the gate, and that will trigger the gate to open. But we can also have voltage-gated channels, and we saw these when we talked about excitation, contraction, coupling. We had voltage-gated calcium channels, and those channels open to allow calcium to flow into our cell based on a change in the voltage. And that is what's referring to a membrane potential change. So when the membrane potential changes along our cell's plasma membrane, that's going to cause the opening of these different voltage-gated channels. Now, just hold on a little longer. I'm going to explain membrane potential, but I'll do that in just a couple slides. So just bear with me for a few more slides, and we'll get into what membrane potential is. But these two are the two types of channels we're going to see the most readily. We also do have mechanically gated channels. These are channels that are opened due to some type of mechanical stimuli, some type of physical stimulus that moves or opens those channels physically. But we're not going to see the mechanically gated channels playing a role in the action potential that we'll cover in this video. Most of the channels we're going to see are the chemically gated and the voltage gated channels. So based on the presence of some type of stimulus, whether that be a neurotransmitter for our ligand gated channels, or perhaps a membrane potential change for our voltage gated channels. Either or, when one of those stimuli is present, that's going to cause the opening or closing of different channels. And when that stimuli is present, that's going to cause the opening of our membrane channels. So once our membrane channels have opened, that's when the electrochemical gradient is going to come into play. And once our membrane channels open, we know that things will flow from a high concentration to a low concentration. And that's where we're going to start seeing our electrochemical gradient. So on this slide, I'm going to further explain the electrochemical gradient. Now, I mentioned this very briefly on our lecture video for Chapter 3, but then we focused primarily on the chemical gradient. So just as a refresher, the chemical gradient is any difference in the concentration of different areas. One area has a high concentration, one area has a low concentration, and the gradient is going to point from a high concentration to a low concentration. So the chemical gradient refers to a difference in the concentrations of different places. Now, the electrical gradient is very similar, but this just refers to the difference in the electrical charges that can be found on either sides of the cell membrane. So what we can hopefully remember from our Chapter 2 lecture video is that positively charged molecules are attracted to negatively charged molecules. And when positive molecules are on one side of the membrane and negative molecules are on the other side of the membrane, those two molecules are going to be attracted to each other and be pulled towards the membrane and want to move towards different sides. So the electrical gradient is the other characteristic that plays into the electrochemical gradient. And both of these gradients combined give us this electrochemical gradient that we're going to use when we understand action potentials. And based on the opening of these different ion channels, certain things are going to be allowed to flow down that electrochemical gradient. So just one more review, we have our leak channels. These are our passive channels, so these may or may not be gated, and we will see these very commonly found throughout our bodies. We see these all over our different neurons in the dendrites, cell bodies, axons, and so forth. But remember, the two that we're really going to be focusing on are the ligand-gated channels and the voltage-gated channels. The ligand-gated channels are going to open in response to some type of ligand that's binding to a receptor. A ligand is just any type of neurotransmitter or other chemical that is binding to a receptor. So when that chemical binds, that's going to cause the opening of our channel and allow certain things to flow down their electrochemical gradient. So we saw these in the membrane of our muscle fiber. Remember, we had ligand-gated sodium channels. Acetylcholine was the ligand, and it came in and allowed for sodium to flow through. So that's one example of a ligand-gated channel. And voltage-gated channels are going to open due to some type of change in membrane potential, which is just a charge difference across the membrane. We'll often see these in the axons, and we saw these in Chapter 10 as well. These were located in the axon, and they were also located in the sarcolemma of our muscle fiber. Mechanically gated channels are just going to open due to some type of physical deformation or some type of touch stimulus. So it's the actual touch itself that forces the channel open. Okay, so that will take us into resting membrane potential. We're going to finally define this membrane potential and start to make some sense for why this membrane potential is so important in terms of sending action potentials out. So the resting membrane potential can be defined as the difference between electrical charges from one side of the cell's membrane to the other. And of course, we call it the resting membrane potential because this is the membrane potential of our cell at rest. When our cell's doing other things like sending out action potentials, this might change. But a resting membrane potential refers to our cell at rest when it's nice and relaxed. So that's really all this resting membrane potential is a difference of electrical charges on one side of the membrane to the other. So due to negative ions building up inside of our cell and positive ions building up outside of our cell, that sets up a charge difference across the membrane. Let's say, for example, we have 50 negative charges built up on the inside of the cell and 50 positive charges built up on the outside of our cell. Our charge difference is going to be a total of 100 charges. In order for us to equalize either of those sides, we would need to add 100 charges to make those sides more equal, whether they be positive charges towards the inside or negative charges towards the outside. Regardless, the difference between that cell would be 100 charges. Now, membrane potential is going to be measured in millivolts, so we'll use these units when we're talking about it. But what we can see oftentimes in neurons is our resting membrane potential is going to be between negative 40 millivolts to negative 90 millivolts. And all that this is telling us is that the difference in the electrical charges on the inside of our cell compared to the outside of our cell is going to be between negative 40 millivolts and negative 90 millivolts. So on average, in a typical neuron, we will say that our neuron most often will have a negative 70 millivolt resting membrane potential. Certain neurons can have different membrane potentials based on their actions, but most neurons are going to have their membrane potential right around negative 70 millivolts. So unless I'm specifically asking for this range on the test, you can assume that all neurons that I ask about are going to be typical neurons and their resting membrane potential will be negative 70 millivolts. That just means that there's a charge difference of negative 70 between the inside and the outside of our cell. And we say that this is negative 70 millivolts because on the inside of our cell we have our negative charges and the outside of our cell is going to be where we find our positive charges. So that's what this negative sign means. It just means that the inside of our cell is negative relative to the outside of the cell. So remember, resting membrane potential was one of the requirements for firing action potentials, and the other was different membrane channels. So now that we've explained the different types of membrane channels, and we've gone over a little bit of what resting membrane potential refers to, we can start getting into how this action potential is going to fire and what that means for our cell. The last thing I want to mention on this slide is that when our cells do have some type of membrane potential, we call those cells polarized. Now, I also mentioned briefly a few minutes ago that membrane potential can change if our cell's doing something exciting. And so if we increase this membrane potential, we increase the difference, we're going to call those cells hyperpolarized. And if we decrease this difference, we're going to call those cells depolarized. And that will become a lot more clear in future slides to come. But I just want you to understand that when I call cells polarized, it just refers to those cells having some type of resting membrane potential. Now you might be wondering why we have a resting membrane potential in the first place. Why do we have a bunch of negative charges on the inside of our cell and a whole bunch of positive charges on the outside of our cell? Now that can be explained on page 419. I do suggest that you go give this a read. I definitely want to make sure that you are comfortable with the information on this page, but I'm going to go through the highlights from the information on that page as well. So there are three things that allow resting membrane potential to exist in our cells. Those are the unequal distribution of ions in our extracellular fluid and the cytosol. This is telling us that we have a difference in concentrations of certain particles on the inside of our cell versus the outside of our cell. And we discussed this very briefly in chapter 3 as well. Potassium concentration is higher on the inside of the cell, and sodium concentration is higher on the outside of our cell. This was discussed in chapter 3. We have these different gradients that need to be maintained, and this is why those gradients need to be maintained. We have to have these gradients in order to give us this resting membrane potential. And we're going to use this resting membrane potential to fire signals. When we start changing the membrane potential around, that's going to help to fire signals and action potentials down the axon. Another reason that we have this resting membrane potential is because anions are often unable to leave our cell. There's a lot of different types of anions, and anions are some type of ion that has a negative charge to it. So the more negative charges that we have on the inside of our cell, the more negative the inside of our cell becomes. So it's very important that we can understand the difference between adding positive charges and adding negative charges. If the inside of our cell is more negative than the outside of our cell, and we add more anions to the inside of our cell, that's going to further increase that membrane potential. That difference is going to become much greater. However, if we add positive charges to the inside of our cell, that's going to make the overall inside of our cell more positive and eventually decrease our membrane potential, because the inside of our cell and the outside of our cell are going to have a much lower difference between the inside and the outside of our cell. So because there's more negative charges and anions that are hanging out on the inside of our cell, that makes the inside of our cell overall more negative relative to the outside of our cell. Now, the third entity that allows us to have a membrane potential is the sodium-potassium pump. So we're really pulling up a lot of this information that we discussed in Chapter 3. Remember, the sodium-potassium pump is our type of primary active transport. And this exports 3 sodium ions out of the cell for every 2 potassium ions that it imports into the cell. So while there's always going to be some leakage across our cell membrane, the sodium-potassium pump maintains these 2 major gradients that we see. So the sodium-potassium pump is maintaining these gradients. So this is indirectly helping to set these gradients up to give us this first reason that we have resting membrane potential. However, the sodium-potassium pump will also directly help give us resting membrane potential as well. So think about the ratios of sodium and potassium that are traded between the inside and the outside of our cell. For every molecule of ATP that is hydrolyzed, we export 3 sodium from our cell and import 2 potassium into our cell. Now, both sodium and potassium ions are assigned a positive charge. Both of these have a +1 charge. So another way that we can look at this is every time we hydrolyze 1 molecule of ATP, we're exporting 3 positive charges and importing 2 positive charges. And if we have lots and lots and lots of these sodium-potassium pumps, we're constantly going to be pumping 1 more positive charge out of our cell than we are bringing into our cell. And that is because of the ratios of trade-off. 3 sodium leave, and that's 3 positive charges, and 2 potassium will enter. But in terms of the electrical difference, these are no different because these both have the same positive charge. So every time this runs, we lose 3 charges from our cell, and we only bring back in 2. So overall, as the sodium ATPase is pumping these 2 ions in and out of the cell, every time this runs, we're actually losing more positive charges from the inside of our cell. So this unequal trade-off of 3 sodium to 2 potassium molecules for every ATP that's hydrolyzed is also helping us to set up the resting membrane potential. As we pump out more positive charges than we bring in, we're making the outside of our cell more positive. So these are the 3 factors that set up our resting membrane potential, and the sodium-potassium pump will eventually be important for maintaining these gradients as well. We're going to come to see that sodium and potassium channels are going to open up, and that's going to disturb all of this membrane potential. And the sodium-potassium pump is what's going to maintain those gradients and restore those gradients back to normal. So this slide is pretty much saying everything that I've just told you in the last slide. But what it shows you in this picture is that we have these positive charges that are lining up along the outside of our cell membrane, and we have these negative charges that are lining up along the inside, because these positive and negative charges are attracting each other through the membrane, and that's what's setting up this resting membrane potential. So if you're still struggling with resting membrane potential, I've posted this video for you here, so you can feel free to go give this a watch if this is still a concept that's not very comfortable to you, but there's also information on page 419 as well. So we've defined resting membrane potential. We know that that's a difference in charges from the inside and the outside of our cell. But we also have the element of our ion channels, and those are going to allow certain things into the membrane and certain things out of the membrane that will cause deviations from that resting membrane potential. So when certain charged particles, let's say sodium for example, flows down its concentration gradient towards the inside of our cell, that's going to overall make the inside of our cell less negative. However, the inside of our cell that's more negatively charged, if potassium ions were to flow out of our cell, that would make the inside more negative. And again, this is because potassium and sodium are both assigned a positive charge. So when we make the inside of our cell less negative, we call that a depolarization. And when we make the inside of our cell more negative, we're going to call that a hyperpolarization. And the reason that we name each of these as such is because in the inside of our cell we have our more negative environment, and so when we decrease that negativity on the inside, we call that a depolarization, because the difference will now be less as it was from the inside to the outside of the cell. Now frankly, action potentials and graded potentials are just deviations from the resting membrane potential. That's all that these two things refer to. That's what these signals are. When our neurons change their resting membrane potential to a depolarized state, that's going to end up firing signals down the axon to eventually go and affect other cells. So all of this information down here is more so referring to graded potentials. So don't let this confuse you. This is not referring to action potentials. This is just referring to graded potentials right here. Now the stuff up here, this is true for both action potentials and graded potentials, but I just wanted to make that clear. I'll probably go ahead and post a different version of the slides with this updated, but I just wanted to make that clear for this video that this stuff down here is just referring to the graded potentials. So let's define graded potentials and then we'll really break down the action potential. So graded potentials have a decrease in their magnitude the further that they travel, and that's why these can only travel a short distance. Action potentials can travel over a much longer distance and their magnitude does not decrease with distance. And that is because our axons have the myelin sheath, which will insulate the axon in order to keep that signal in and strong. Now we can also have a difference in the magnitude of our graded potentials. Graded potentials will eventually converge on one neuron and summate, and that's going to eventually determine whether or not our neuron is going to fire an action potential. So graded potentials are needed in order to fire action potentials. So this is a big slide. I definitely want you guys to understand the concepts on this slide. Again, just remember that the stuff below this line is all going to be referring to graded potentials specifically. So the graded potentials are going to fire similarly to the action potentials, but remember, those just occur from the dendrites to the cell body. This signal is going to make its way, and I should actually say a whole bunch of these signals are going to make their way, and they're all going to be converging on that axon hillock. And when our neuron takes all these graded potentials and consolidates them, it will then be able to decide whether or not it's going to fire an action potential or not. So an action potential can be defined in one way as the rapid decrease in membrane potential, which is also called a depolarization, and then an immediate repolarization, where the membrane potential is restored to the resting membrane potential. So that's really all an action potential is a depolarization followed by a repolarization. We're deviating from our resting membrane potential, and then we're immediately bringing our membrane potential back to its resting state. So the action potentials are going to be a little bit different than the graded potentials. Their magnitude's not going to decrease over distance because we have those myelin sheaths insulating our axon and keeping that signal in. Again, the action potentials will be fired down an axon, while graded potentials will be signaled from the dendrites to the axon hillock. And those graded potentials will be coming in from many different neurons. We're going to get lots of different signals, and those are going to converge at the axon hillock. And so some of those potentials are going to be positive, some are going to be negative, and they'll eventually summate to allow our cell to decide whether or not it's going to fire an action potential on its own. But truly, what we're trying to understand about the action potential is we're just having a depolarization followed by an eventual repolarization. We're going to make the inside of our cell much less negative, and then we're going to bring it back to its resting state. That's really what an action potential is referring to. So this picture shows us what an action potential would look like on a graph. Now on this particular graph, we see time on the x-axis and we see voltage on the y-axis. Now voltage is in millivolts, which we know is our units of our resting membrane potential. Now we can also see that this right here is the resting state. This is at negative 70 millivolts, and that's where a typical neuron is going to have its resting membrane potential. So some type of stimulus is going to excite our neuron. That's going to cause a depolarization. Now again, at negative 70, our cell is at its resting membrane potential. So when we make the inside of our cell less negative, That's going to decrease the difference in the charges from the inside to the outside. Because remember, the outside is positively charged and the inside is negatively charged. So if we decrease the negatively charged inside to a more positive place, that's decreasing the difference between the inside and the outside. And that's why we call this a depolarization. And an action potential is just a brief reversal of membrane potential. So the depolarization is going to occur, and then our membrane potential is going to increase all the way back up to the resting state and actually beyond. We call this the refractory period, where no other new action potentials can be fired from that particular neuron. We have to eventually bring ourselves back to the resting state before we can fire another action potential. We call this part of our graph where the membrane potential has actually become more negative than -70. That's going to be the hyperpolarization, where we are now much more negative on the inside of our cell. And that's why we have this refractory period. And we'll make a little bit more sense of this as we go in the different events that occur during depolarization, repolarization, and hyperpolarization. But for this picture, I just want you guys to get an idea of the brief reversal of events and how this is going to take place. So again, just so we can go through this picture, at the resting membrane potential, some type of stimulus is going to cause a deviation in our membrane potential to cause a depolarization that brings us all the way to actually a more positive membrane potential. And eventually this is going to be met with an immediate rapid repolarization that will eventually go past the resting membrane potential and be referred to as a hyperpolarization. And then this will eventually bring us back to our resting state, and we can fire another action potential. Now there's one other element of this picture that I want to explain, and that is this threshold found at -55. So what's going on with this threshold here? The threshold potential is the required deviation in order for our cell to fire an action potential. And that's why we see these failed initiations here in the center. These are failed initiations. Certain stimuli have caused these initiations, and they've caused little deviations in our membrane potential. However, none of these deviations were strong enough to reach us to the threshold potential. So in order for us to fire an action potential, we have to reach the threshold potential, which is at -55 millivolts. So if a stimulus can depolarize our resting membrane potential enough to the threshold potential, we'll fire an action potential every single time. However, if we cannot reach -55, if we had another stimulus here that brought us to -56, that would not be strong enough and we would not fire an action potential with that stimulus. That is referred to as the all or none principle, and I'll explain that in much greater detail in a little bit. But right now, I want us to take some time to understand what's going on with these different channels during depolarization and during repolarization. So at the resting state, our sodium and potassium channels are closed. There's not a lot of trading off of these ions from the inside and the outside of our cell. Now I want you to keep in mind that sodium and potassium will both be assigned a positive charge. So when either sodium or potassium moves in or out of the cell, that's going to change the membrane potential. The difference in charges is going to change with those two ions that are flowing in or out of our cell. So at the resting state, our resting membrane potential is more negative on the inside of our cell. And we'll also see a higher sodium concentration on the outside of our cell and a higher potassium concentration on the inside of our cell. ECF just stands for extracellular fluid, ICF stands for intracellular fluid. So potassium is in higher concentration on the inside, sodium is in higher concentration on the outside. This is how I've been explaining cells since chapter 3. We have a higher sodium concentration on the outside of our cell and a higher potassium concentration on the inside of our cell. So this is how our cell is existing at its resting state. However, when we undergo a depolarization, some things are going to happen. So the major event that occurs during our cell's depolarization is the opening of voltage-gated sodium channels. So in order for these voltage-gated sodium channels to open, we have to have some type of depolarization from an outside stimulus to -55. We'll discuss the threshold in just a little bit, but right now I just want us to understand the different stages of the action potential. So when something brings our cell down to -55 millivolts, the cell itself is going to finish depolarizing the cell. So if sodium-gated channels open in our cell membrane, that's going to allow sodium to flow down its concentration gradient. Sodium is high on the outside and low on the inside. So sodium will flow towards the inside of our cell. So keep in mind, every sodium ion is going to have a positive charge. So if we open up sodium channels, that's going to bring a lot of positive charges towards the inside of our cell and change the membrane potential. Through this depolarization, the inside of our cell is becoming much less negative because we're adding a bunch of positively charged sodium ions towards the inside of our cell. So that's why when we see this picture, if we're depolarizing our membrane potential, this is showing us that sodium channels are open inside of our cell membrane, and that's allowing sodium to flow into the cytosol of our cell. So this is the major event that occurs through depolarization, is the opening of voltage-gated sodium channels. Now, action potentials are reversals in membrane potential. So we'll have a depolarization that's followed by a repolarization, and the repolarization is then going to be followed by a hyperpolarization. So let's go through each of these states as well. So with a repolarization, the major events that occur are our sodium gates closing and potassium gates opening. Our sodium gates need to close to stop any sodium from flowing into our cell, and that's what's going to stop the depolarization. However, at the same time, our potassium channels are going to open. So if potassium channels are opening, we need to understand which direction potassium is going to flow. Potassium is high on the inside of our cell and low on the outside of our cell. So potassium is going to flow towards the outside of our cell. So basically, in these first two steps, we've traded the sodium and potassium from the inside to the outside of our cell. And at this point, after repolarization, potassium is now in a much higher concentration on the outside of our cell. And the reason that this restores our resting membrane potential is because potassium also has positive charges assigned to it. So sodium flew in, which made the inside less negative, and then potassium is going to flow out to make the inside much more negative than it had just become. And this is actually why we will have the refractory period, why we can't fire another action potential, is because we need to restore these sodium and potassium concentrations on the inside and the outside of our cell. So during repolarization, our voltage-gated sodium channels close and our voltage-gated potassium channels are opening. And that's allowing for sodium to stop flowing towards the inside of the cell, and that's also going to allow for potassium to flow out of the cell as well. Now during our hyperpolarization, the potassium channel gates are not going to abruptly close. They're actually going to close rather slowly, and we see that shown right here. You can see that there's not necessarily as abrupt of a change as we saw up here. This was a very, very brief reversal that occurred. And here, this is going to be represented by our potassium channels that are slowly closing. And so again, until we can restore our sodium and potassium concentrations on the inside and the outside of our cell, we're not going to be able to fire another action potential. So up here, this is where we see hyperpolarization. So during our hyperpolarization, the net charge of the inside of our cell is much more negative than it is at its resting state. And our membrane is what is referred to as hyperpolarized. Now eventually we need to be able to restore these sodium and potassium gradients, and that is going to be done by the sodium-potassium pump. Every 3 sodium that's exported, 2 potassium will be imported, and that's how we can restore these sodium and potassium concentrations back to our resting state. So I know that this slide looks a little frightening because there's a lot of words on it, but I promise that this slide just goes over the concepts that we've discussed on the last couple slides. So at the resting state, at our cell's resting membrane potential, we have sodium and potassium ions that are not going through the membrane. So at our resting state, when our cell is at its resting membrane potential, we'll have sodium in high concentration on the outside of our cell and potassium at high concentration on the inside of our cell. And we'll also have our resting membrane potential at negative 70 millivolts. This is telling us that we have a greater negative net charge on the inside of our cell relative to the outside of our cell. Once some stimulus has brought us to the threshold potential, that's going to cause a depolarization. And the major action of depolarization is the opening of sodium channels. This is allowing sodium to go through our cell membrane towards the inside of our cell, down its concentration gradient, and that results in a depolarization, which is just referring to making the inside of our cell less negative. And after these sodium channels open and our depolarization occurs, we're going to have the abrupt closing of these sodium channels, and then the opening of our potassium channels to allow us to have our repolarization. The repolarization stage is characterized by potassium ions flowing out of our cell towards the extracellular fluid. And those potassium ions with a positive 1 charge leaving the cell are going to make the inside more negative. Because if we lose positive charges from the inside of our cell, and those positive charges go towards the outside of our cell, that makes the inside of our cell more negative because we lost positive charges. This repolarization stage will be followed by the hyperpolarization stage, which is just denoted by the slow closing of our potassium channels. And eventually, in order to restore those concentrations of sodium and potassium on the outside and the inside of our cell, we're going to use the sodium-potassium pump to trade 3 sodium and 2 potassium between the membrane. This is occurring during the refractory period, also known as hyperpolarization. And lastly, once we have restored these concentration gradients on either side of our cell membrane, we're going to be ready to fire another action potential. So action potentials will trigger at the axon hillock, and they will travel down the axon and eventually go and affect whatever cells are attached at the axon terminals. Or I should more accurately say synapsing at the axon terminals. Now this picture is again showing us our action potential. However, in this version, we've color-coded this. So each of these colors depict certain events that are occurring with our sodium and potassium channels. So in this purple color we see at the beginning and the end of our action potential, we see that this is showing us our resting membrane potential. Our voltage-gated sodium channels are closed and our voltage-gated potassium channels are closed. So everything is normal in this purple color on either side. However, when we get to this blue color, this is showing us the depolarization to the threshold potential. Some type of stimulus needs to get us to the threshold potential, and that is shown with this deviation in the membrane potential up to -55. However, once our cell reaches the membrane potential of -55, or that threshold potential, the cell is going to be able to self-activate its own action potential, and that is done with the opening of our voltage-gated sodium channels, and that can be shown in green. So depolarization is characterized by the opening of our voltage-gated sodium channels. After this, we have our immediate reversal into the repolarization phase. And the repolarization phase is characterized by the opening of potassium channels and the closing of sodium channels. And this is what allows us to bring our membrane potential back to this -70. So eventually, in the hyperpolarization phase, this is when our voltage-gated potassium channels are still open and they're closing very, very slowly. And the sodium-gated channels are still going to remain closed. So these potassium channels are closing very, very slowly, and that's allowing for potassium to continue to flow out of our cell past our resting membrane potential. And then that sodium-potassium pump is going to restore our membrane potential back to its resting state. And once we're in this resting state, once again, we'll be able to fire another action potential. So our repolarization is always going to chase the depolarization. We have a brief reversal of a depolarization followed immediately by a repolarization. So this is the action potential. This is how neurons communicate with other neurons and muscle cells. This is what the action potential is the change in the voltage difference across the membrane. That's what our resting membrane potential is showing us. So I'd like you to feel comfortable with the events that occur during each stage of the action potential. And I'd also like you to feel comfortable with the different channels that open up and the trading out of ions between the inside and the outside of our cell. Now, I know that this is a big process, so if there are any questions about this, always feel free to send me an email. I'd be happy to spend a little bit of extra time with you discussing how this process works if you're really struggling with this. So this slide is just kind of reiterating some of the concepts that we've learned so far. In order for communication to occur between cell to cell, our action potential must fire from the axon hillock all the way down to the axon terminals and through the synapse into our next cell. Now, unlike graded potentials, action potentials will not lose their magnitude over time. And this is all thanks to the myelin sheath that is insulating our axon of our neuron. So this slide will further go over the threshold potential. Now we're going to discuss the threshold potential in the next video as well when we get into graded potentials. But for now, I want you to understand that the threshold potential is the membrane potential required in order for our cell to fire an action potential. So you can see on this picture that we have 3 different stimuli: a subthreshold stimulus, a threshold stimulus, and a suprathreshold stimulus. And the only difference in these is their magnitude and how long they occur. So the subthreshold stimulus is any type of stimulus that is not strong enough to bring our membrane potential to that threshold potential. This is going to cause a deviation in the membrane potential that's not quite to the threshold. So this is not going to fire an action potential. Our membrane potential will change, but we're not going to have the depolarization, repolarization change that we saw in the last few slides. However, a threshold stimulus is any stimulus that's strong enough to depolarize our cell to -55. And once our cell is depolarized to -55, the neuron itself is going to take over and propagate the rest of that action potential. And so that's going to be showing us this depolarization followed by a repolarization. Now, a suprathreshold stimulus, of course, is going to be a lot stronger and a lot longer than normal threshold stimuli. But what's important to note is regardless of how strong the stimulus is, that's not going to change the magnitude or the intensity of our action potential itself. All of these action potentials have the exact same intensity. These action potentials are just occurring more frequently because the time of our suprathreshold stimulus is going to be much longer than our threshold stimulus. So this is referred to as the all or none principle. Our cell either fires an action potential or it doesn't fire an action potential, and this is solely based on whether or not we've reached the threshold potential or not. So if we can depolarize to that threshold potential at -55 millivolts, that's going to fire our action potential. If we make it to -56, that will be a sub-threshold stimulus and our cell will not fire an action potential. That is the all or none principle, which will explain our threshold potential and why we need to have it. And it's actually quite useful that we have this threshold stimulus, because if we didn't, and any stimulus could cause a depolarization that would fire an action potential, we would be firing action potentials all the time. When we get into graded potentials, you'll see that there are constantly going to be deviations in our cell's resting membrane potential. There's always going to be depolarization, and we want to only fire action potentials when our threshold stimulus has been met. So there are some factors that will affect how quickly action potentials will propagate down our axon. And those are axon diameter, temperature, and the amount of myelination that's present in our myelin sheath. So let's go over each of these three. So the axon diameter is the first factor that's going to affect our propagation speed. The larger the diameter of the axon, the less resistance that's going to be present traveling down that axon. There's going to be a much larger surface area for our signal to propagate. So the larger the diameter, the faster our action potential will fire. If we have a very, very small axon, that signal might travel a little slower. With temperature, as we will most often see, the higher the temperature, the faster the action potential will fire. Most things, when we increase the temperature, will move a lot faster, and the action potential is no different. And lastly, the amount of myelination. Myelination is just our insulation formed by our Schwann cells in the peripheral nervous system. The more myelin we have, the more insulation we have, and the faster that action potential is going to propagate. And this slide is showing us the difference between the speed of an axon that's myelinated versus the speed of an axon that's unmyelinated. So what I want you to see here is that the neuron on the left side is unmyelinated, and the neuron on the right side is myelinated. You can see these Schwann cells wrapping around. This is our myelin sheath, and on this axon, there's no myelin sheath at all. This is going to be a much slower signal, and this is going to be a much faster signal because we have this myelin sheath wrapping around and insulating this axon. So you can see we have three different versions of this neuron. One's at 1 millisecond, one's at 5 milliseconds, and one is at 10 milliseconds. And the same times are shown for each of these. But you can see with the first millisecond that each of these signals are going to be starting in the axon hillock. And as time progresses to 5 milliseconds, these action potentials are going to go further and further down the axon. And you can see that still at 5 milliseconds they're fairly the same, but at 10 milliseconds this unmyelinated axon hasn't really sent its action potential very far. However, with this myelinated neuron, you can see that with 10 milliseconds, we're almost to the end of our axon. So this slide is just to show you that myelination allows our action potentials to propagate much faster.